If you are listening to this, you have probably found yourself in a debate or, at least quietly listening to one about what is quote unquote natural when it comes to human beings.
Oh, yeah, constantly.
Right, because we love to put things into neat little boxes. We crave it. Like, think about a medical diagnosis. You break your arm, the x-ray shows a jagged white line, and the doctor just points to it. Broken or not broken, it's a binary, and it's incredibly comforting to have that level of certainty.
It is. And, from a psychological standpoint, that craving makes perfect sense.
Yeah.
We are fundamentally pattern-seeking animals. The human brain burns a massive amount of calories just trying to navigate ambiguity.
So we just look for shortcuts.
Exactly.
Yeah.
Whenever we can slap a label on something like male or female, monogamous or promiscuous, natural or unnatural, our neurology essentially rewards us. It just feels safe to have a rigid category.
But the deeper you look into the actual machinery of life, the more you realize that X-ray machine is, well, it's completely broken. When you step out of the high school biology classroom and look at the sprawling reality of evolutionary biology, genetics, and neuroscience, those rigid categories just disintegrate.
They absolutely fall apart.
And that is exactly our mission for this deep dive. We are going to bypass the political shouting matches, drop the cultural baggage, and look exclusively at the raw data. We are taking a sledgehammer of hard science to the pseudoscientific myths surrounding sex, gender, monogamy, and human nature.
And we're going to be unpacking a massive amount of research today to do that. We are pulling from comprehensive university lectures on the neurobiology of sexual behavior, alongside two deeply researched biological analyses that completely dismantle the idea of a strict biological sex binary.
It's a lot of material.
It is, yeah. But my goal here is to explain the actual mechanisms at play. Not just what happens, but why it happens. Because once you understand the cellular and evolutionary logic, how the machinery truly operates, the old myths just fall apart on their own.
And we are not pulling punches today. The biological consensus right now is clear, and frankly, the way outdated science is constantly cherry-picked to justify dogma and cruelty is something we have zero tolerance for.
Right. It's just bad science.
Exactly. So let's start with a myth that almost all of us grew up with. I'm talking about the 1950s nuclear family ideal. There is this incredibly pervasive assumption that lifelong, exclusive monogamy, one man, one woman paired for 60 years with zero deviation, is the ultimate hardwired natural law for humans.
Right. It's a beautiful romantic ideal, but when evolutionary biologists actually analyze the human species, that assumption hits a massive brick wall.
Really? Why is that?
Well, to understand why, you have to look at how biologists categorize mating strategies across the animal kingdom. They generally divide species into two major evolutionary buckets. You have your tournament species and your pair bonding species. And the biological hardware required for each strategy is, it's radically different.
Okay, break that down for me. What exactly are the mechanics of a tournament species? Because when I hear tournament, I'm picturing like some kind of brutal gladiatorial combat.
Honestly, that is actually not far off. Think of gorillas or baboons or elephant seals. In a tournament species, the evolutionary strategy is all about fierce male-to-male competition for reproductive access.
So they fight for the right to mate.
Exactly. And because of this, their physical bodies evolve specific traits. The primary one is extreme sexual dimorphism, meaning the males and females look entirely different.
Like size-wise?
Yeah, size, weapons, all of it. Male gorillas are massive, sometimes twice the size of females. They develop huge canines, heavy musculature, thick skulls. They are literally built for violence.
Because the prize for winning that violence is genetic immortality.
Right. In these societies, mating is highly skewed. The data shows that in many tournament species, about 90% of the mating is done by the top 5% of the males.
Wow. 90%.
Yeah, it's a winner-take-all, hyper-aggressive system. And because the dominant male is spending all his energy fighting off challengers, he provides absolutely zero parental care to the offspring. He just passes on his genes and moves on to the next fight.
Okay, so brute force, massive physical differences between the sexes and absent fathers. That is the tournament model. On the complete opposite end of the spectrum, we have the pair bonding species. What does their hardware look like?
A classic example of a pair bonding species would be the marmoset monkey, or certain species of voles and birds. In these species, the evolutionary strategy relies on cooperation to ensure the survival of the offspring.
So they aren't fighting to the death for harems.
Exactly. And because they aren't doing that, you don't see that massive sexual dimorphism. Males and females are roughly the exact same size. They don't have giant defensive weapons like massive canines.
Right, they don't need the biological armor because they aren't going to war every mating season.
Precisely. Instead, they form stable, long-term partnerships. And the parental investment is entirely egalitarian. Like in marmosets, for instance, females frequently give birth to twins.
Oh, wow. That sounds exhausting for a small monkey.
It is. The caloric demand of nursing twins is so high that the mother would literally starve if she had to forage and carry them alone. So the biological father is deeply involved. He carries the infants, protects them, shares food. It is a highly cooperative monogamous system.
Okay, so we have the ultra-aggressive polygamous gorillas on one side and the egalitarian monogamous marmosets on the other. Now, if you were listening to this, you were probably trying to figure out which bucket humans fall into.
And this is where the data gets incredibly awkward for anyone trying to claim humans are biologically hardwired for strict monogamy.
Awkward how?
Because if you look at every single physiological metric, humans fall perfectly, messily right in the middle. We are a biological compromise.
Give me an example of that compromise in our actual bodies.
Let's look at body size dimorphism. Remember, gorillas are double the size, marmosets are the same size. Human males are, on average, about 10 to 15% larger than human females.
So we aren't double the size, but we aren't identical either.
Right. Then look at testes size, which evolutionary biologists use as a primary indicator of sperm competition.
Wait, explain that connection. How does test size indicate mating behavior?
Well, in a promiscuous species where a female might mate with multiple males in a short window, the males aren't just competing physically, right? Their sperm is competing internally to fertilize the egg.
Oh, it's a microscopic race.
Exactly. And to win that microscopic race, you need sheer volume. So species with high sperm competition evolve massively large tests relative to their body weight to produce an overwhelming amount of sperm. Chimpanzees, which are highly promiscuous, have enormous tests.
And gorillas.
Gorillas, where the dominant male has exclusive access and zero sperm competition, have incredibly small ones.
So where do humans rank on the sperm competition scale?
Right in the middle. Larger than a gorilla's, smaller than a chimp's. When you look at our body size, our canine teeth, our reproductive organs, the physiology paints a very clear picture. We are neither entirely biologically polygamous nor entirely biologically monogamous.
We're an intermediate species carrying evolutionary traits for both strategies.
Exactly. We are built for a bit of both.
You know, there was a piece of data from the Stanford Neurobiology lectures that just floored me. They looked at global marriage and divorce rates across various traditional and historical human cultures, not just modern Western societies, but globally.
Yeah, that data is fascinating.
It is, and they found that the average duration of a marriage or a pair bond before it dissolves or shifts to a new partner is remarkably consistent. It's usually 2 to 4 years.
Which is an incredibly specific and revealing time frame.
Right, because that 2 to 4 year window exactly correlates to the average interbirth interval in traditional human hunter-gatherer societies.
Exactly. The interbirth interval is basically the time between a mother giving birth to one child and being biologically ready to give birth to the next.
Okay, so how does that connect to the 2 to 4 year bond?
Well, in traditional cultures, without, modern baby food or safe sedentary environments, a child requires maximum heavily dependent parenting for about 3 to 4 years. During that time, the mother is often nursing, which suppresses ovulation, and the infant is entirely reliant on the parents for survival.
So the evolutionary logic here is that we form a pair bond just long enough to get this incredibly vulnerable, big-brained human infant through its most perilous stage of development.
Yes. And once the child is slightly more self-sufficient, that biological imperative to stay bonded begins to wane.
That makes so much sense.
The anthropological evidence strongly suggests that humans are naturally adapted for serial monogamy. We are wired to form a deep pair bond, raise dependent offspring through that critical 3 to 4 year window, and then, very often, we experience a biological inflection point where that bond dissolves and we seek out a new genetic partner.
So the physiological drive to remain with one single person for 60 years with zero temptation is simply not supported by our evolutionary hardware.
No, it's really not.
But what is actually happening in the brain when that bond forms and when it breaks? The research on voles completely changes how we think about love and faithfulness. Scientists looked at polygamous voles and monogamous voles, and it all boils down to the mechanics of 2 neurochemicals, right? Vasopressin and dopamine.
Yeah, this is where we get to see the actual machinery of monogamy.
Right.
It centers heavily on dopamine, specifically how it interacts with two different receptors in the brain, the D1 and D2 receptors.
Okay, how do those work?
Think of a receptor as a lock, and dopamine is the key. In monogamous voles, when they first meet a mate with them, and form a pair bond, there is a massive flood of dopamine that primarily hits the D2 receptors.
And what does the D2 receptor actually do?
D2 receptors mediate the anticipation of reward. It is the thrill of the new bond, the intense infatuation phase. It is what drives the animal to want to be near this specific partner.
Honeymoon phase.
Right, the honeymoon phase. But here's the critical part. Once that bond is firmly established, the brain actually undergoes a structural shift. It downregulates the D2 receptors, meaning it makes them less sensitive, and it significantly upregulates the D1 receptors.
So the brain literally changes its own wiring once the relationship is secure. D2 forms the relationship, but D1 maintains it.
Precisely. High levels of dopamine activating those D1 receptors mediate the faithfulness of the bond. When D1 is active, the vole is highly satisfied with its current partner, and it actually becomes aggressive or completely indifferent toward any new, unfamiliar voles that try to mate with it. It locks the bond in place.
And the proof of this is wild. In lab experiments, researchers took perfectly monogamous voles that had been bonded for life. They introduced a chemical that selectively blocked just the D1 receptors. What happened?
The monogamous behavior completely vanished. Within about 8.5 minutes of decoupling the D1 receptor, the vole would abandon its lifelong partner and happily mate with a new stranger.
Wait, 8 minutes?
Just 8 minutes. The biological hardware for faithfulness was simply turned off.
8 minutes to erase a lifelong pair bond. It really puts human behavior into perspective. It's like our hardware is trying to run two conflicting operating systems at the exact same time.
That's a great way to put it.
Right. Because on one hand, we have this craving for stability, safety, and D1 receptor maintenance. We want the warm, secure bond. But on the other hand, we have this evolutionary baggage from our tournament species ancestors, which drives what biologists call the Coolidge effect.
Yes, the Coolidge effect. That is the biological phenomenon where a male animal, even if sexually exhausted with his current partner, will suddenly experience a massive surge of arousal and stamina if introduced to a novel, unfamiliar female.
It's just the raw evolutionary drive for genetic variety.
Exactly.
So we are walking around with the neurological capacity for deep, enduring love, while our ancient reptilian brain is constantly scanning the horizon for new genetic opportunities. No wonder human relationships are so complicated.
Oh, absolutely.
But before we move on, I want to challenge a piece of that tournament species narrative. We talked about gorillas and baboons relying entirely on brute force and aggression, but the data shows that even in the most violent tournament species, the biggest, meanest alpha male doesn't necessarily win the evolutionary game, do they?
No, they don't, and this was a massive blind spot in early evolutionary biology.
Really? How so?
For decades, male scientists observed these primates and assumed it was a simple linear access model. The strongest male beats up all the weaker males, and therefore he automatically gets all the females.
It sounds like a very human assumption to make.
Exactly. But when researchers actually started paying attention to the female's behavior, the narrative completely collapsed because female baboons exhibit active choice.
And who are they choosing? Because it isn't always the alpha.
No, they frequently choose to mate with what the primatologists literally categorized as nice guys.
Nice guys.
Yes. These are lower-ranking males who refuse to participate in the bloody, exhausting dominance battles. Instead of fighting, these nice guy baboons spend their time engaging in affiliative behaviors. They groom the females, they share food, they sit quietly and play with the female's infants.
Oh, wow. So they're essentially building social currency instead of physical dominance.
Exactly. So when the alpha male is having a violent, screaming tantrum, the nice guy is sitting quietly grooming a female. And when that female goes into estrus and is ready to mate, she will very often sneak off into the bushes, completely bypass the alpha male, and choose to mate with the nice guy who has been kind to her.
That is incredible. It proves that female choice, empathy, and social cohesion are deeply rooted evolutionary strategies. Survival of the fittest doesn't just mean survival of the most violently aggressive.
Right. Sometimes it means survival of the most emotionally intelligent and cooperative baboon. It completely shatters this toxic idea that hyperaggression is the only quote-unquote natural male state.
It really forces us to recognize that social bonds and empathetic behavior have immense evolutionary value, even in species we previously categorized as pure brutes.
Yeah, it changes everything about how we view animal societies.
All right, so we have dismantled the 1950s monogamy myth. Now, let's turn our attention to the absolute core myth of this entire discussion. We are taking that sledgehammer to the high school biology lie.
Oh, this is a big one.
You know the one. The idea that if you have XX chromosomes, you are 100% female, and if you have XY chromosomes, you're 100% male. End of story. Check the box on your birth certificate, no exceptions.
That is perhaps the most pervasive and ultimately damaging oversimplification in modern society. The reality, as any geneticist or endocrinologist will tell you today, is that biological sex is a massive, multi-layered spectrum. It is absolutely not a binary.
I want to start with the statistic from the biological analyses we reviewed because it fundamentally shifts the scale of this conversation. Differences in sexual development, or DSDs, which were formerly referred to as intersex traits, occur in roughly 1.7% to 2% of live human births.
Right, and let's put that number into perspective. 2% of the global population is roughly 130 to 136 million people.
That's huge.
It's equivalent to the entire population of Russia.
But look, if I am a skeptic listening to this, I am going to look at that 2% statistic and say, okay, sure, 2% of people are born with biological variations, but mathematically that's just a tiny anomaly, a rounding error. Why should we rewrite the entire biology textbook and redefine male and female just to accommodate an exception?
That is a very common pushback, but it betrays a fundamental misunderstanding of how science works. In science, an exception doesn't mean you just sweep the data under the rug and pretend it doesn't exist.
Right.
An anomaly is a signal that your underlying rule is fundamentally flawed. If your model cannot account for the data, you don't throw away the data. You build a better model.
It reminds me of the analogy the authors used about the periodic table.
Oh, it's a brilliant comparison. Look at the observable universe. 99.9% of all atoms in the universe are either hydrogen or helium.
Which is a massive, overwhelming binary.
Exactly. It makes the 98% human sex binary look entirely insignificant. But you would never look at a chemist and say, well, because 99.9% of the universe is hydrogen and helium, we should just ignore carbon, oxygen, gold, and iron. They're just anomalies, so let's throw away the rest of the periodic table.
Right. You don't call gold an anomaly and pretend it doesn't exist. Those other 116 elements exist. They follow the laws of physics. They're entirely natural.
Exactly. If your biological model of human sex cannot seamlessly account for 136 million living, breathing human beings, then your model is broken.
Right.
The human beings are not broken. The rigid binary framework we force them into is broken.
So let's build the accurate model. The biological consensus breaks human sex down into 4 distinct chronological steps or layers. It is not just one master switch flipped at conception. It is a cascading biological sequence. We have chromosomal sex, gonadal sex, anatomical sex, and phenotypic sex. And the wild part is they rarely align perfectly.
Let's walk through the mechanics of those steps. Step 1 is chromosomal sex. This is established at the exact moment of fertilization when the sperm meets the egg. Usually this results in an XX or XY pairing.
And step 2.
Step 2 is gonadal sex. Around week 6 of fetal development, specific genes trigger the undifferentiated tissue in the fetus to become either ovaries or testes.
Got it. And step 3 is anatomical sex. This is where the internal ducts and the external genitalia form, and this step is driven heavily by the hormones that those newly formed gonads start pumping out.
Right. And finally, step 4 is phenotypic sex. This encompasses the secondary sex characteristics that develop much later during puberty, things like breast tissue growth, facial hair, vocal changes, and fat distribution.
Okay, so the flawed assumption of the binary model is that if you start as XY at step 1, you just perfectly slide down a frictionless chute and automatically hit male on steps 2, 3, and 4.
Exactly. But biology is incredibly chaotic, and interference can happen at every single one of those steps. Let's look at the very first step, chromosomes. We assume XX and XY are immutable laws, but they are subjected to a phenomenon called gene hopping.
I need you to explain this because the idea of genes just jumping around sounds completely chaotic. Why does this happen?
To understand gene hopping, you have to understand a process called meiosis, which is how our bodies produce sperm and egg cells. During meiosis, chromosomes line up next to each other and intentionally swap chunks of genetic material.
Okay, why do they do that?
This process is called crossing over. And it is an evolutionary mechanism designed to increase genetic diversity so that you aren't just an exact clone of your parents.
Oh, so it's genetic shuffling.
Exactly. Now, the primary gene responsible for initiating male gonadal development, the gene that says build testes, is called the SRY gene. Usually this gene sits safely on the Y chromosome, but during that chaotic genetic shuffling process of crossing over, mistakes happen. Sometimes the SRY gene accidentally breaks off the Y chromosome and hijacks its way onto an X chromosome.
Which leads to a condition called Dulé-Chappelle syndrome.
Yes. In Diehl-Chappelle syndrome, an individual inherits an X chromosome from their father that has the SRY gene attached to it, alongside a normal X chromosome from their mother. So their genetic blueprint is completely XX.
They are chromosomally female.
Right. But because of that one relocated SRY gene, step 2 is triggered. They develop tests, they produce testosterone, and they develop male external anatomy.
So they are chromosomally female, but anatomically and phenotypically male. And this process works in reverse too, right?
It does. If a father's Y chromosome loses its SRY gene during the swap, he passes on a Y chromosome that is essentially missing its male programming switch. The child will have XY chromosomes, but they completely lack the genetic trigger for testicular development.
And this is called Swyer syndrome.
Yes. Consequently, these individuals develop female internal and external anatomy.
And the most paradigm-shifting detail about Swyer syndrome is that these individuals, people with XY chromosomes, can carry a pregnancy and give birth to healthy children. You literally have people with male chromosomes giving birth.
Yeah, it's pretty incredible.
If that doesn't completely vaporize the high school biology binary, I honestly don't know what does.
It proves definitively that having an X or a Y chromosome is neither necessary nor sufficient for determining your overall biological sex. It is just one piece of a vast puzzle. And if you think gene swapping is wild, wait until we look at the cellular level. Let's talk about chimerism.
This is the phenomenon that honestly sounds like a sci-fi movie. How does a human chimera form?
Chimerism occurs very early in a pregnancy, when a mother is carrying fraternal twins, 2 separate eggs, fertilized by 2 separate sperm. Occasionally, those 2 early clusters of cells will merge together in the womb to form a single embryo.
Whoa! So the resulting person is literally a patchwork of 2 completely different genetic blueprints.
Yes.
So if one of those original twins was XX and the other was XY, what happens to the surviving person?
Their body becomes a mosaic. Some of their cells, organs, or tissues might have XX chromosomes, while the cells sitting right next to them have XY chromosomes. You could have a person with ovarian tissue on the left side of their body and testicular tissue on the right. Their blood cells might be one sex, while their skin cells are another.
That is mind-blowing, and it gets even more common with something called microchimerism. I had never heard of this before, but it completely broke my understanding of the human body. What is happening between a mother and a fetus?
During a normal pregnancy, the placenta acts as a barrier, but it is not completely sealed. Fetal stem cells frequently cross the placenta and enter the parent's bloodstream. These stem cells are pluripotent, meaning they can transform into almost any type of tissue. If a chromosomally female mother is carrying a chromosomally male child, those XY stem cells can travel through her bloodstream, cross the blood-brain barrier, and implant themselves into her organs.
And they just survive there.
For decades, researchers have found intact, functioning XY cells living inside the livers, hearts, and brains of women in their 70s who gave birth to sons 50 years prior.
Wait, seriously?
Yes. Think about the implications of that for a second. If you are a mother to a son, you could literally have cells of a different biological sex beating inside your own heart or processing thoughts inside your own brain right now.
It beautifully and profoundly illustrates that even at the most fundamental microscopic cellular level, the human body is a diverse mosaic. It refuses to be a uniform monolithic binary. All right, so we've established that step 1, chromosomes, is messy. Let's move to step 3, hormones and anatomy. There is a very common pop science myth that I hear repeated constantly. It is the idea that all human fetuses start as females. We all start as girls, and then the Y chromosome drops in and masculinizes us. But the biological data explicitly corrects this, doesn't it?
Yes, that is a vast and fundamentally incorrect oversimplification. Up to about 6 weeks of fetal development, fetuses are in what developmental biologists call the indifferent stage.
Indifferent stage.
Right. They are not female. They are entirely sexually undifferentiated. They have a single urogenital opening called a cloaca. And critically, they possess two distinct sets of internal ducts simultaneously.
The blueprints for both options.
Exactly. They have the Müllerian ducts, which have the potential to develop into the uterus, fallopian tubes, and upper vagina. And sitting right next to them are the Wolffian ducts, which have the potential to develop into the vas deferens, epididymis, and seminal vesicles.
So how does the body decide which blueprint to build?
It is an incredibly complex, aggressive genetic tug-of-war. Sex differentiation isn't just about initiating one pathway. It requires actively destroying the other.
Destroying it.
Yes. For example, if the SRY gene is present, it triggers another gene called SOX9. SOX9's job is to build testes. But to do that successfully, SOX9 must actively suppress a counter gene called WNT4, which is constantly trying to build ovaries. It is a biological battle.
Wow.
The testes, once formed, secrete anti-Müllerian hormone to literally dissolve the female ducts while simultaneously pumping out testosterone to develop the male ducts.
But what happens when there is a miscommunication in that hormonal signaling? Let's talk about CIS, complete androgen insensitivity syndrome, because this shows how hormones can completely overload chromosomes.
CIS is a fascinating condition regarding cellular communication. In a person with CIS, their chromosomal sex is XY. Their gonadal sex develops as tests, and those internal tests produce very high, typical male levels of testosterone.
Okay, so they have the male blueprint and the male hormones.
However, their physical cells possess a genetic mutation that alters the shape of their androgen receptors.
I think of this like a radio station. The tests are the radio tower broadcasting a testosterone signal at absolute full blast. But because of this mutation, every single cell in the body has a broken antenna. The signal is filling the airwaves, but the body physically cannot receive it.
That is a perfect analogy. Because the body cannot perceive or bind to the testosterone, the masculinization process completely stalls. The external genitalia develop entirely along the female anatomical pathway.
So they appear female.
Yes. These individuals are born looking physically female, they are assigned female at birth, and they are raised as girls. In fact, many individuals with CAIs have absolutely no idea they possess XY chromosomes or internal tests until they reach puberty and fail to menstruate, prompting a medical scan.
Their phenotypic reality is completely divorced from their chromosomal reality. And then you have an inverse phenomenon happening with a community in the Dominican Republic with children locally known as the Gavedoses.
Right, this is one of the most remarkable naturally occurring variations in human biology. In this specific isolated community in the Dominican Republic, there was a high prevalence of a genetic deficiency regarding an enzyme called 5-alpha reductase.
Okay, what does that enzyme actually do in a developing fetus?
Well, during fetal development, the testes produce testosterone. But standard testosterone isn't potent enough to shape the external male genitalia, the penis and the scrotum.
It needs a boost.
Exactly. The body needs to convert that testosterone into a much stronger hormone called dihydrotestosterone, or DHT. The 5-alpha reductase enzyme is the chemical machine that performs that conversion.
So these children have XY chromosomes, they have tests, and they produce standard testosterone. But because they lack that specific conversion enzyme, they can't make the highly potent DHT.
Right. And without DHT in the womb, the external genitalia do not masculinize. These children are born with external anatomy that appears female. They are assigned female at birth and are raised socially as girls. But then, biology throws a massive curveball at puberty.
The puberty hormone surge.
Yes. When these children hit puberty around age 12, the body initiates a massive secondary surge of standard testosterone.
Yeah.
This surge is so overwhelmingly powerful that it essentially brute forces its way past the enzyme deficiency.
Yeah.
Even without DHT, the sheer volume of testosterone triggers the dormant developmental pathways. Their internal testicles descend, they literally grow a penis, their voices drop, and their muscle mass increases.
The local slang, gavedoses, literally translates to testicles at 12.
Yes.
And what is so fascinating about the anthropological data here is that these individuals transition into living phenotypically and socially as men within their culture, and it's largely accepted. Their chromosomes never changed, their genes never changed, but their anatomical and phenotypic sex radically transformed through entirely natural biological mechanisms.
It's incredible. And when you synthesize all of this data, the crossing over of chromosomes, the cellular chimerism, the mutated receptors, the enzyme deficiencies, the scientific conclusion is undeniable. Biological sex is bimodal, meaning there are two main physiological peaks that most people fall into, but it's fundamentally a vast, multi-layered, fluid spectrum bridging those peaks. It is a complex landscape. not a rigid binary line.
All right, if human biology is this sprawling and complex, I want to pull the lens back even further. What about the rest of the wild kingdom? Because one of the primary arguments you hear from people trying to enforce strict gender roles is that being distinctly male or female is just nature's law.
Oh, if you look at the broader animal kingdom, nature doesn't just bend the rules of sex, it breaks them with staggering chaotic enthusiasm. The diversity of sex determination across different species makes the human XXXY system look incredibly boring and rigid.
I want to explore some of these because the mechanisms are mind-blowing. Let's start with reptiles.
Okay, so many reptiles, such as leopard geckos and sea turtles, don't use sex chromosomes at all. Their genetic code doesn't dictate their sex. Instead, they rely on temperature-dependent sex determination.
Wait, how does temperature literally rewrite an animal's anatomy?
It comes down to how environmental heat interacts with specific enzymes inside the egg. In leopard geckos, the temperature of the sand they are incubated in activates or deactivates an enzyme called aromatase.
And what does aromatase do?
Aromatase is responsible for converting androgens, which are male hormones, into estrogens, the female hormones. If the sand is incubated at an intermediate temperature, say 30 degrees Celsius, the aromatase is highly active, flooding the embryo with estrogen, and the gecko develops as female.
Okay.
But if the temperature rises to 32 degrees, the heat literally denatures and turns off the aromatase. The androgens take over, and the gecko becomes male. If it gets even hotter, up to 34 degrees, it switches back to female. The physical temperature of the dirt dictates their sex.
That is wild, the temperature of the dirt. What about insects? Let's talk about ants, because their family tree logic completely broke my brain.
Ants, bees, and wasps use a system called haplodiploidy. In this system, the queen mates and stores sperm. When she lays an egg, she has a choice. If she releases sperm and fertilizes the egg, that egg develops into a female worker or a new queen, possessing a full set of chromosomes, half from the mother, half from the father. This is the diploid part.
But what if she just lays the egg without fertilizing it? How does an unfertilized egg even survive?
That's the haploid part. The unfertilized egg just begins dividing anyway, using only the single set of chromosomes from the mother. And every single one of those unfertilized eggs automatically develops into a male.
Which creates a biological paradox, because if a male ant only comes from an unfertilized egg, it means a male ant has no father. And because he can only pass his sperm to a queen to create female workers, a male ant can never have a son. He can only have a grandson by passing his genetics through a female daughter. The entire concept of human lineage and biological legacy just simply doesn't apply to them.
It's an entirely alien evolutionary strategy. And when we look at marine life, it gets even more fluid. Take the slipper limpet, which is a type of sea snail. They are sequentially hermaphroditic, and their sex is determined entirely by physical contact and social environment.
How does that work?
Slipper limpets live in stacks, literally piling on top of each other on the ocean floor. When a larva lands on an empty rock, it develops as a female. When a second larva lands on top of her, the physical, chemical signals of touching another limpet cause the new arrival to develop as a male.
So they just build a tower of alternating sexes.
Basically, yes. But here's the crazy part. If another male lands on top of the pile, the males sandwiched in the middle will undergo a rapid hormonal cascade and physically transition into a female. Their sex is dictated entirely by their real estate and who they are touching.
That is insane. And we see social triggers in larger fish too, right?
We do. The stoplight parrotfish lives in coral reefs, and they operate in harems with one dominant male. If that dominant male dies or disappears, the largest female in the harem doesn't go looking for a new male.
What does she do?
Her brain senses the social absence, triggers a massive hormonal shift, and she literally morphs into a fully functioning reproductive male to take over the harem.
Wow. But out of all the examples, the absolute most staggering one from the research was the fungi.
Oh, yes. The split-gill mushroom. While human society fiercely debates whether there are two biological sexes, the split-gill mushroom operates with over 23,000 distinct mating types.
23,000. How does a species even manage that biologically?
It uses an incredibly complex genetic lock and key system. Instead of just X and Y, they have two genetic regions, let's call them A and B. Region A has hundreds of variations, and Region B has roughly 100 variations. The combination of those variations results in 23,000 unique genetic identities.
And they can all mate with each other.
Almost all of them. The evolutionary brilliance of this system is that any single mushroom can successfully mate and exchange genetic material with 22,999 of the other types. The only type it absolutely cannot mate with is its exact genetic duplicate.
It is a biological mechanism purely designed to prevent inbreeding and maximize extreme genetic diversity. When I hear all of this, it makes me realize that trying to force nature into a neat, binary little box is like looking at a wild, sprawling Jackson Pollock painting and trying to paint a rigid, black and white, paint-by-numbers grid over it. It just doesn't fit. Nature is a spectrum of chaos, fluidity, and boundless creativity.
It is, and recognizing that breathtaking diversity is essential to actually understanding biology,
Right.
rather than just imposing our narrow cultural biases onto the natural world.
Which perfectly brings us to part 5, the leap from the physical body to the brain and the crucial distinction between sex and gender. We have spent this entire time proving that biological sex, the hardware of the body, is a massive fluid spectrum. So how do the researchers define gender?
The scientific consensus is very precise in separating these two concepts. Sex refers to the biological characteristics we've been discussing, the chromosomes, hormones, gonads, and anatomy. Gender, however, refers to a combination of socially constructed roles, behaviors, and most importantly, a person's deeply held internal neurological sense of identity.
Okay, so it's a separate system.
Exactly. While sex is the biological hardware, gender encompasses the software how an individual's brain experiences and expresses their identity within a cultural framework.
Now, I want to address a major point of confusion here. A lot of people struggle with this distinction. They will say, look, your brain is a physical organ. It is part of your biology. Therefore, your internal gender identity must just be automatically dictated by whatever chromosomes you were born with. But the neurobiology of fetal development completely refutes that assumption.
It does, and it all comes down to developmental timing in the womb. The sources highlight a biological timing discrepancy that is absolutely vital to understand if you want to grasp the reality of being transgender.
Let's get into the timing.
During fetal development, the sexual differentiation of the genitals, the physical formation of the penis or the vulva, happens relatively early. It primarily occurs during the first trimester, roughly between weeks 6 and 12.
Okay, so the body gets its architectural instructions early on. When does the brain get built?
Well, the basic structure of the brain forms early, but the sexual differentiation of the brain, the intricate structuring of neural pathways and behavior centers influenced by massive washes of hormones, happens much, much later. That process typically occurs in the second half of pregnancy, deep into the third trimester.
So you have two completely separate developmental windows occurring months apart.
Precisely. And because these processes happen at completely different times, under entirely different hormonal conditions, and influenced by different genetic expressions, they can easily take different paths.
Wow.
From a purely biological, neurological standpoint, this perfectly explains how a person can be born with the physical anatomy of one sex, but possess the brain structure and internal gender identity of another. The two systems developed independently of one another.
And we can actually see evidence of this in brain scans, can't we? The researchers cited fascinating studies focusing on a specific tiny region of the brain called the bed nucleus of the stria terminalis, or the BSTC. Before we get to the findings, what does the BSTC actually do?
The BSTC is a region in the brain deeply involved in regulating anxiety, stress responses, and social behavior. Neuroanatomists have known for a long time that the BSTC typically exhibits size dimorphism between males and females, meaning it generally has a different volume and neuron density in typical male brains compared to typical female brains.
So what happened when they looked at the brains of transgender individuals?
The postmortem studies were incredibly striking. They examined the brains of transgender women individuals who were assigned male at birth, based on their anatomy, but who deeply identified and lived as women.
And what did the scans show?
The researchers found that the size, volume, and neuron density of their BSTC perfectly matched the typical female brain, not the male brain. Their neuroanatomy aligned with their gender identity, not their physical birth sex.
And the crucial control variable here is hormone therapy, right? Because skeptics might argue that taking estrogen physically altered their brains.
That is the most powerful part of the findings. The researchers found this exact structural alignment regardless of whether the individual had ever undergone hormone replacement therapy or gender-affirming surgeries. Their brains were structurally organized to match their internal gender identity from the beginning, completely independent of their assigned sex at birth.
Which completely vaporizes the political argument that being transgender is just a psychological delusion, a choice, or a quote-unquote social contagion. The actual physical hardware of their brain matches their identity. And speaking of the idea that gender is just a choice or something you are socialized into, we need to talk about one of the most tragic case studies in modern medical history. the David Reimer case.
It is a profoundly heartbreaking story, but it serves as the definitive, tragic proof against the idea that gender is merely a social construct that can be forced onto a child. In the 1960s, an infant boy named David Reimer suffered a horrific medical accident. During a routine circumcision, the equipment malfunctioned, and his anatomy was irreparably destroyed.
That's just awful.
It was. His terrified parents sought out the top experts in the country, and they ended up consulting a psychologist at Johns Hopkins named John Money.
We need to understand John Money's mindset at the time, because he was operating under a very specific psychological theory.
Yes, the 1960s was the era of radical behaviorism. Money was a massive proponent of the blank slate theory. He vehemently believed that gender identity was entirely a social construct. In his view, biology didn't matter at all. If you took an infant, dressed them in pink, gave them dolls, and told them they were a girl, their brain would simply accept it, and they would become a girl.
So Money saw David's tragic accident as the ultimate opportunity to prove his theory.
He convinced David's parents to take a radical step. They had David undergo sex reassignment surgery as an infant. They put him on intense estrogen regimens as he grew. They changed his name to Brenda, and they raised him strictly, unequivocally as a girl.
And Money published papers on this, right?
For years. He published papers touting the experiment as a massive success, claiming it proved socialization dictates gender.
But the reality was a catastrophic, agonizing failure.
From the very beginning, David resisted. Despite being treated exclusively as a girl by his family, his school, and society, David experienced severe, paralyzing gender dysphoria. His internal, hardwired neurological gender identity remained resolutely male.
He knew who he was.
Yes. He tore off his dresses. He felt completely alienated from his own body. Eventually, the psychological toll became so unbearable that he threatened severe self-harm if he was forced to continue visiting Dr. Money and living as a girl.
And when his parents finally broke down and told him the truth about his past when he was a teenager, what did he do?
He felt an overwhelming sense of relief that he wasn't crazy. He immediately transitioned back to living as a man. He took testosterone. He underwent surgeries to reconstruct a male chest. He married a woman and adopted her children. But the trauma of that forced socialization haunted him his entire life, and tragically, he ultimately died by suicide in his late 30s.
David's tragic life is the definitive scientific proof that you cannot peer pressure, socialize, or force a gender identity onto someone. The internal sense of gender is deeply, structurally hardwired into the brain. You cannot raise a boy to be a girl, and you cannot bully, legislate, or therapy a transgender person into being cisgender.
Which is exactly why the global scientific and medical consensus universally condemns conversion therapy. Attempting to force someone to change their deeply held gender identity or sexual orientation is not just medically ineffective, it is actively, devastatingly harmful. It causes severe psychological trauma. The authors of our research refer to it as pseudoscientific cruelty, and the historical and clinical data overwhelmingly supports that conclusion.
Now, we are moving into the final major section of this deep dive, where the rubber meets the road, science versus dogma. And it is important to clarify something here for you listening. The researchers and biologists who authored the papers we are discussing take a very sharp, uncompromising stance on how this science is currently being manipulated in our political and cultural climate. We are impartially exploring their central thesis, but we stand absolutely uncompromisingly by the biological data they present.
Their central thesis is stark. They argue that political charlatans, reactionary commentators, and certain dogmatic institutions intentionally weaponize a fake, cartoonish version of nature.
How do they do that?
They cherry-pick the oversimplified high school biology. We just spent the last hour dismantling the strict XXXY binary, the hyper-aggressive tournament male, and they use it as a cudgel to justify transphobia, homophobia, and the systemic marginalization of anyone who doesn't fit into a very specific, rigid Western framework.
They use fake biology to enforce cruelty. And what is so insidious is that it is not just biological complexity they are erasing, it is human history. The authors highlighted how modern dogma relies on rewriting the past, specifically pointing to the myth of man the hunter.
Oh, this is a classic one.
We all know the stereotype. Big, strong cavemen went out and hunted the mammoths to provide, while the weak, passive women stayed back at the camp, gathered berries, and watched the babies. It is constantly used today to justify modern patriarchal gender roles as our evolutionary destiny.
But the archaeological and ethnographic data completely obliterates that narrative. The authors point to a massive recent survey of indigenous and foraging societies globally. When anthropologists actually looked at the burial sites, the bone structures, and the historical practices, they found that women actively participated in hunting big game in 79% of the surveyed cultures.
79%. That is not an anomaly, that is the overwhelming norm.
Exactly. Women were out there tracking, running down, and killing large animals alongside the men. In burial sites in the Andes, they found female skeletons buried with the exact same big game hunting toolkits as the males. The biomechanics of human endurance running are shared almost equally between sexes.
So the man-the-hunter narrative is not a scientific reality.
Not at all. It is a modern 1950s cultural bias projected backward in time to justify contemporary inequality.
And that intentional erasure extends powerfully to gender identities around the world. The researchers detail how Western colonialism systematically wiped out or marginalized beautiful, complex, non-binary gender identities that had existed for centuries in indigenous cultures.
This is a vital historical context. The strict, inflexible gender binary is largely a Western colonial export. Prior to colonial suppression, hundreds of indigenous cultures globally recognized, integrated, and often revered third, fourth, and fluid gender identities.
Can you give some examples?
Sure. The sources point to the Mahu in traditional Hawaiian culture, who embody both male and female spirits. They point to the Hijra in India, a recognized third gender with deep historical roots. They point to the diverse two-spirit identities across numerous Native American tribes in North America.
These weren't considered anomalies or outcasts either. These individuals often held high social and spiritual regard. They were healers, mediators, and visionaries.
Right. The rigid binary was forced onto these cultures through violence and assimilation, intentionally erasing centuries of magnificent human diversity.
It proves that acknowledging gender fluidity is not some new modern trend. It is the historical baseline of humanity. The strict binary is the recent invention. And the hypocrisy surrounding how we enforce that binary today, specifically regarding how we police people's bodies, is astounding. The researchers dissect the current moral panic surrounding gender-affirming care. We see politicians passing laws to ban gender-affirming care for transgender people, claiming it is unnatural, dangerous, or a mutilation of the body. But when you look at the clinical data, a massive, undeniable hypocrisy emerges.
It is perhaps the most glaring double standard in modern medicine. If you look at the vast majority of medical procedures and prescriptions that fall under the exact umbrella of gender-affirming care, things like hormone replacement therapies, testosterone supplements, estrogen pills, breast augmentations, breast reductions, facial reconstruction, and hair transplants, the overwhelming majority of these procedures are actually consumed by cisgender people.
Think about that. Cisgender men in their 50s taking testosterone supplements because they don't feel physically manly enough. Cisgender women undergoing painful surgeries to get breast implants to align their physical bodies with their internal sense of femininity. Cisgender men getting hair plugs to fight off baldness and preserve a youthful, masculine appearance.
Exactly. When a cisgender person uses modern medical science to alter their physical body to better align with their internal gender ideal, society embraces it. We call it self-improvement, anti-aging, or just standard healthcare.
But when a trans person does it...
Right. When a transgender person seeks the exact same medical interventions using the exact same hormones and the exact same surgical techniques to alleviate the severe documented distress of gender dysphoria, it is suddenly legislated against and labeled as a dangerous mutilation.
It is not about the medicine. It is about who is allowed to have agency over their own body. And the data on the psychological outcomes is the final nail in the coffin for these bans. The researchers point out that banning this care for transgender people is a total rejection of the global medical, psychological, and pediatric consensus.
The clinical data on outcomes is overwhelmingly positive. When you track regret rates for gender-affirming transition procedures, they are astonishingly low, typically hovering around 1%.
1%?
Yes. And to put that into medical perspective, the regret rate for a common knee replacement surgery is roughly 20%. The regret rate for LASIK eye surgery is higher than the regret rate for gender transition.
So when we pull all of this together, from the complexities of our chromosomes to the evolutionary realities of our ancestors, from the intricate wiring of our brains to the clinical data on medical outcomes, what is the central thesis we are left with?
The primary takeaway from all of this biological research synthesizes into a single, scientifically grounded call for empathy. The universe is complex. Biology is chaotic, fluid, and endlessly diverse. Using bad, cherry-pick science to control, isolate, and legislate against vulnerable people is not an intellectual oversight. It is an intentional choice to be cruel.
Yeah.
Real intelligence, both scientifically and morally, requires us to update our understanding when presented with undeniable evidence. When the biological data unequivocally shows that sex, gender, and human nature are vast, beautiful spectrums, the only rational and humane response is to accept it.
Which brings us to the end of our deep dive today. We set out to take a sledgehammer to the myths, and the science did the heavy lifting. We have seen that from the gene-hopping chromosomes of de la Chapelle syndrome to the temperature-changing leopard geckos, from the 23,000 distinct sexes of a split-gill mushroom to the intricate, independent neuroplastic pathways of the human fetal brain. Biological reality is messy, it is spectacular, and it absolutely refuses to be stuffed into two tiny, rigid boxes. Understanding the full, chaotic spectrum of our biology doesn't break our understanding of humanity, it profoundly, deeply enriches it.
We are part of a wildly diverse natural world. Our survival and our strength as a species lie in our ability to understand that complexity, adapt to it, and celebrate it, rather than fear it.
I want to leave you with one final provocative thought to mull over. Throughout this deep dive, we touched on the concept of neuroplasticity, the scientifically proven fact that living a specific life, experiencing the world in a specific way, and interacting with your environment actually wires and shapes the physical, structural pathways of your brain. Your experiences literally build your brain.
They do.
So if that is biologically true, consider this. How much of human potential have we literally stunted on a structural neurological level by forcing billions of developing brains to conform to a rigid, artificial gender binary for the last few centuries? How much anxiety, depression, and suppressed brilliance have we engineered into our society by demanding that people fight against their own deeply hardwired nature? What kind of minds, what kind of innovations, and what kind of societal brilliance might we develop when we finally step back, drop the dogma, and let human nature be exactly as wonderfully complex as it actually is?
It genuinely makes you wonder what humanity is truly capable of when we stop fighting a war against our own biology.
Exactly. It's time we stop trying to read a blurry, oversimplified X-ray and just look at the magnificent, complex person standing right in front of us.